Thank you so much for joining us today. It is such a pleasure to welcome Rachel McMinn, CEO of Neurogene, and Stuart Cobb, Chief Scientific Officer. As many of you know, Neurogene announced in mid-May that they have added a Rett syndrome gene replacement program to their company drug development pipeline. This was extremely welcome news to Rett families in the US and around the world. Now, many of you are already familiar with Stuart Cobb. He's been working on Rett syndrome for over 2 decades, or about 2 decades. His first paper on Rett was in fact the famous 2007 Rett reversal paper from Adrian Bird's lab, and Stuart was a co-author on that. RSRT has funded Stuart's lab at the University of Edinburgh for many years, and we are so pleased to see his gene replacement program transition to a company. I first met Rachel McMinn in early 2018. She visited me at home and told me about her plans to start a company focused on devastating genetic diseases. And after meeting her, I had no doubt in my mind whatsoever that Rachel was going to do just that. And it is such a pleasure to now introduce her to the broader Rett community. So welcome, Rachel. Welcome, Stuart. Thank you so much for taking the time out of your extremely busy schedule to join us. Now, before we get started, just a comment about Q&A. We anticipated that there would be a lot of questions, and in the interest of trying to answer as many as possible and as thoroughly as possible, we solicited questions in advance. So once our conversation with Rachel and Stuart has wrapped up, I will ask them the questions that we've received. So Rachel, we're going to start with a question for you. Please tell us a little bit about your background. What inspired and compelled you to start Neurogene?
Well, first, I just want to say thank you, Monica, and thank you to everybody for dialing in. It is a July 4th weekend, and I'm sure everyone has plans. So let me tell you a little bit about myself. I'm a PhD scientist by training. After doing that and doing some additional work after my PhD, I decided that I really wanted to be involved with science, but just at a different level, not necessarily in the lab. And I transitioned to Wall Street as an equity analyst, which means I made stock recommendations and followed biotechnology stocks very, very closely for over 13 years. And when doing that, I found myself towards the tail end of my career with a lot of very strong opinions about how a company can be successful and, what are the hallmarks of an unsuccessful company. And, to be honest, I, loved my job. I was very good at it, but I really felt like I wanted to do something more than just, make money for investors. And I thought about actually founding my own company. I didn't go straight to that. There's a big difference between being on the outside doing analysis and being on the inside and actually leading. So I went to a liver disease company, trained there for several years, about 3.5 years. But I really found myself asking, like, why am I in liver disease? What does this really mean? I wasn't emotionally connected. And this is where the personal side of my story really matters. And I really wanted to make a big difference in the world about something that I really cared about. And for those of you who haven't, gone to our website, my older brother is severely neurologically impaired. And I grew up with a lot of dysfunction in my family. And I kept thinking about, like, why are there no treatments for him? And I knew, especially having been on the science side, having been on the business side, that the brain is just so complicated and so poorly understood that it's just a therapeutic area that's riddled with failures. But I, was sort of undaunted, decided that this is something that mattered to me. I started digging into potential ways to create a company focused on these types of devastating neurological diseases. And as part of my research, I came across a paper that many of you may be familiar with, but it's about a gene replacement called Zolgensma for the treatment of babies with SMA or spinal muscle atrophy. And when I read the paper, it was just this crystallizing moment for me, that genetic medicine could have such a profound, really profound impact on these kids and on their families. And me being me, 2 months, I didn't waste any time. 2 months after I read that paper, I founded Neurogene. And I really just, felt in my bones that I wanted to be part of this genetic medicine revolution. We're still early days in that, and we'll talk a little bit about that. But I just felt like when I lined everything up with all of, my training in science and business and my personal connection with my brother, if I didn't start a company, then what resume of a person would actually start this kind of a company? So a little bit of a long answer, but at least gives you a sense of why, what the why is behind Neurogene.
Thank you, Rachel. And we're grateful that your brother inspired you. I mean, what a legacy for your brother. So tell us just really succinctly what Neurogene's mission is.
Yeah, I mean, it's pretty simple. We want to turn devastating neurological diseases into treatable conditions. And so if you think about, to me anyway, cancer like 50, 100 years ago, you got cancer, you died. And now, people can live with cancer for decades and actually have a reasonably high quality of life, depending on the kind of cancer. And so I think that, we want to make the quality of life, not just extending survival, but actually making people actually feel better. And that's really our mission, to do the same for neurological diseases.
And what was it about 2018 that made it the right time to start the company?
Well, it's a collision of, just circumstances, like most important things in life. So as you heard, like, I was actually personally ready to do it. And so obviously the company wouldn't exist if that weren't true. But it was also a time where, there was a lot of enthusiasm around gene therapy. And as a result, there were a lot of investors that were not only willing to fund, but they were willing to fund at very early stages in the innovative process. And that hadn't been true when I first started on Wall Street. People were much more cynical. And so I think those are important factors. And the last thing is just to say, I had a very strong reputation as a, kind of very diligent person on, with investors. And so as a result, I had a lot of people that invested in Neurogene because they believed in me, frankly. And so I think, the sort of that compilation of factors made it all possible.
That's great. And as we know, Stuart had been working on Rett for many years before you started Neurogene. Can you tell us a bit about how you met him and how did he become your CSO?
Well, it's a really great story. In my research on various diseases, I did become, and I've told you this in subsequent conversations, Monica, directly, but I was very attracted to studying Rett syndrome. I'll share with you all that back in the 1970s, when my brother was first diagnosed, he received a diagnosis of autism. And that word is very loaded. It means a lot of things to a lot of people. But in any case, my initial idea for Neurogene was to really focus on autism spectrum disorders, and specifically monogenic disorders that I thought could be more tractable. And, I came across Rett syndrome and I started reading about it. And there was some overlap between my brother and Rett syndrome. And I just sort of, I don't know how to say it, but I felt like very connected. And, as I started reading as many papers as I could, Stuart's name just kept coming up over and over and over again on these really important publications, as you highlighted. And I just thought, like, geez, I can't just email this guy out of the blue. He'll think I'm crazy. So one of the people I did reach out to was Randy Carpenter, who I assume, everybody on this call knows really, really, really well, as the chief medical officer. And at the time, Randy had just moved to Hawaii. So I just decided that, meeting Randy and being able to connect to Stuart somehow was so important that I literally decided I had to meet him in person. I bought myself a plane ticket to Hawaii with the only purpose of seeing Randy. And to this day, I haven't quite figured out why he took the meeting. It was quite funny, but But in any case, we started talking. I told him my story and what I wanted to do. And he just, after several hours had this brilliant idea that I should meet Stuart. And so that's sort of the funny thing. And then he introduced us. We had a phone call. We all got, Stuart, I'm sure you remember that first phone call. Like everyone was so excited. And then we just decided, well, this isn't enough. So then Randy and I flew together to actually go to Edinburgh and meet Stuart and the team. And we spent a couple of days there, met a lot of people at Edinburgh, I also brought with me a blizzard, and have very fond memories of that as well. But so it was really from there, we started a collaboration. We had just such a wonderful scientific exchange of ideas and such a good rapport. It just dawned on me, like, I need to build a really good team. And like, I just believed in Stuart, and I asked him to be my chief scientific officer, and like magically he said yes. So that's kind of the story. So I'm so grateful for the faith that Randy had in me, and our, and the Neurogene vision, and that really helped accelerate our collab and made feasible our collaboration.
Great. I guess it was meant to be. So Stuart, you started off as a basic scientist, right? A scientist who does research for the sake of knowledge, but became a translational scientist, right? On a mission for individuals with Rett syndrome and other neurological disorders. Can you share a little bit about your role as Simon's Fellow and Neuroscience Reader at the University of Edinburgh?
Sure, Monica. So yeah, so I certainly started out as a basic researcher, sort of studying, fundamental aspects of disease. But I moved over to translational research, in other words, applied or therapeutic research, when I set up my own lab. So at the University of Edinburgh, my role is really sort of early stage. It's basically driving the innovation and what's known as discovery stage research. And this includes things such as understanding the disease biology and developing the technologies. And what really differentiates my University of Edinburgh work from work at Neurogene is that the Edinburgh activity is really about exploring the novel technologies, whereas Neurogene's about pulling everything together to actually make a drug.
And being CSO of a company is a major responsibility. What drew you to Neurogene?
Well, being in translational research, ultimately you want the work that you're doing to go to help patients, and universities are great for lots of things, but they don't, create drugs. That occurs within industry. So it was never going to happen in the university lab, and I was interested in partnering with a company, but not any company. So the people in the mission really mattered to me. So when Rachel approached me, as she's just described, I was just sort of so impressed with her reasons for creating Neurogene, and also impressed with the people she was beginning to sort of pull together to form Neurogene. So when she offered me the opportunity to take on this role, I was delighted to accept.
When you started working on Rett syndrome, strategies like gene therapy were still considered, pretty much science fiction, right? Especially gene therapy to the brain. What has it been like, kind of to be a witness as all this progress has happened?
Yeah, I think you're right, Monica. The field of gene therapy has really exploded. So, pretty much all major pharmaceutical companies and hundreds of biotechnology companies are now working in this space of genetic medicine. However, you're right, when we first started on, down the path of a gene therapy for Rett syndrome, the area was still, really quite fresh. And indeed, the field had sort of been scarred with some early safety issues with gene therapy. So really, the development of gene therapy has been quite a roller coaster. It's sort of very positive news one minute and then sort of dashed the next. So it's been very varied. But, you know, overall, it's a hugely exciting time. But I guess it's also important to sort of moderate expectations. Rachel gave the analogy with cancer therapy decades ago. We're still at the early days of gene therapy and genetic medicines. And like all drugs, you know, the first generation is always going to be superseded by better versions as the technology improves and as knowledge improves. And this will undoubtedly be the case for gene therapy as well. But you know what's very clear, and you know, as the example Rachel gave with Zolgensma, we're beginning to see genetic medicines, you know, helping in disorders where there was previously no therapy whatsoever. So this is really a huge advance.
Yep. So Rachel, you know, neurological disorders are typically not low-hanging fruit for companies, and yet they are the focus for Neurogene. So first of all, maybe you can help us understand why it's so challenging to tackle neurological disorders with genetic medicine and actually in general with any kind of medicines, and what makes Rett syndrome particularly challenging.
Yeah, it's a good question. I mean, I think as we kind of talked about earlier, the brain is really the most complex and organ and least understood in the body. And I'm sure that is not a surprise to anybody on this call. What makes gene therapy even more difficult is that, you know, even the best delivery methods, right, like how we actually get this into the patient, you know, even the best approaches really don't penetrate all cells in the brain. And so that's just important, you know, to think about. You know, if you stick some virus, you know, which is, you know, kind of coats the healthy copy of the gene that we're instilling, you know, there's just not the level of precision and amount of control that we'd like to get into every single neuron, as an example. So in some cases, you know, some cells in some parts of the brain get a lot more AAV penetration, while other cells get little or not even any, you know, any of the gene replacement. We refer to this, we call this biodistribution. And the way to think about it, or at least this is the analogy I came up with, you can maybe like it or not like it, but, you know, imagine you have a FedEx truck like that, that's the vector, right? So it's the truck, the packages are inside, and they deliver packages. If you live in New York City, you get your FedEx stuff pretty much on time when they say they're going to. But if you live out way in a small town, very far away in the middle of nowhere, like you might not get your packages for days, or you might not get your package. And so you kind of have this one time you give gene therapy, and you've got a lot of these cells that are pretty far removed from where the gene therapy is. And as a result, you're not going to get into every cell. You know, that's important, because that means that we have this imperfect kind of delivery particle, and we want to get the gene into as many unhealthy cells as possible. So that's sort of the gene therapy concept for the brain, and why we're making progress, but we're not sort of at the pinnacle by any means. As we think about Rett syndrome, you know, out of all neurological diseases, it's even more challenging. Because the MECP2 gene that is dysfunctional in, you know, all of your children that have a diagnosis with Rett syndrome, that gene needs to be very, very tightly controlled. So, you know, in conventional gene therapy, if you just kind of take the current technology that is, you know, we went beyond that, and we'll talk a little bit more about it. But if you just take conventional, you get some cells that just have huge amounts of vector, and as a result, it's very, very toxic. So you can't just use a regular standard gene therapy because it's going to make those, you know, those cells get sick and die. And Stuart's actually shown that very high levels will kill mice as well. So it's not just a theoretical, it's like an actual demonstrated scientific fact that overexpression is toxic. So really being able to get to kind of the right set point, the right levels that, you know, don't cause toxicity, but are still, you know, showing benefit, that's really the key objective in any type of gene replacement Rett syndrome program.
Yeah, thanks, Rachel. That was helpful. And I'll just make a comment in terms of not being able to hit every cell. Certainly true. There is data, fortunately, from animal model experiments in Rett syndrome that we can get improvements even if we don't hit every cell, and that even hitting 3 to 5% of cells does show some improvement. So that's I think the question will be, how good is AAV9 for Rett syndrome? That's a question that remains to be answered.
Absolutely fair point. And I know we're going to get to this later, but that's why there's only so much you can extrapolate from animals at the end of the day. We are not mice, and it'll be important to see how it translates. It may be, like you say, very little goes, has a massive effect, but we just don't know. We don't know.
So Stuart, can you describe for us the investigational gene therapy product that Neurogene is developing for Rett, and how it compares and contrasts with other approaches for gene therapy for Rett?
Sure. So I guess the first thing I would say is that we're still in the preclinical, what's termed the preclinical stage at the moment. So we're basically completing experiments that would be required to open a clinical trial. So that's the stage we're at. But in terms of the actual product, as you sort of alluded to, it's a viral gene therapy. So it uses AAV9 as the delivery vehicle to deliver a functional copy of the MECP2 gene. So that's really the approach. But I guess the spin that we have is that we're delivering it in a controlled way, so that you deliver enough but not too much. So basically, it works by having what's known as a single gene circuit, and you can think of this a bit like a thermostat. So irrespective of how much virus you're delivering, there's a sort of inbuilt thermostat that controls the level of MECP2, so that you can't give or you can avoid giving too much. So it really controls the MECP2 within a safe range, as seen in our animal studies to date. So that's really so this thermostatic approach is one differentiator from what other people are doing, and I guess another differentiator is the fact that we're putting back the full length MECP2 gene, at least the protein coding sequence, what's known as the protein coding sequence of the gene. So MECP2 is a very, very highly conserved protein through evolution. And we know that in Rett syndrome, just changes to a single letter in the alphabet that codes MECP2 can cause Rett syndrome. So we think it's really important to put back, the full copy of the dominant form that's in the brain, expressed in the brain. So that's a key point. And then finally, another, I guess, unique approach with our product is not the product per se, but the way they were delivering the gene therapy. So we're delivering the gene therapy by what's known as ICV gene therapy, which stands for intracerebroventricular gene therapy. So that's delivering it directly into a space in the brain. It's a hollow space known as the ventricle. And we have experimental data to show that that's the most efficient way to, effectively deliver the gene therapy product to the nervous system, and in other words, to hit as many cells as possible and be as corrective as possible. So, basically all of these features are predicted to boost the effectiveness, but also the safety of this product.
Great, thank you, Stuart. So Rachel, as you know, the Rett syndrome community has been waiting a number of years for a gene replacement clinical trial to start. There was a lot of disappointment, myself included, when Novartis terminated their gene therapy program last year. Tell us a little bit about next steps for the Neurogene program. What are the milestones we should be looking out for? And do you have any sense of timeline, which, as you can imagine, that's on a lot of people's minds that are tuning in today?
Yeah, and it's always always disappointing when you kind of think something's coming and it stops, but it does actually speak to the nature of science and medicine, and in particular in gene therapy. But for us, we announced in May in conjunctions, like our data. I think this was a big announcement for us. So this was preclinical data in mice showing efficacy and safety, and we do have we have a series of other animal studies ongoing, and those are not just important, but really critical to complete because they're part of the process. So in order to advance into any clinical trial, there's so many different studies, especially with something like we've talked about with Rett syndrome, where you're going to be cautious and making sure that your treatment is actually safe. So, once those studies are completed, it then takes several months for all the data to be analyzed. So there's that's one component. At the same time, so these are not done, in isolation, but we're also making gene therapy, and gene therapy manufacturing is sort of a whole 1-hour webinar that we could talk about it at some future time. But we do have our own manufacturing in-house. The early results are very encouraging. So we've been able to make this at a scale that is robust and, kind of supporting a clinical trial. But nonetheless, we still have to analyze all of the material that we're making and, complete lots of experiments to really make sure that NGN-401, the product that Stuart talked about, that we understand what the purity levels are, what the potency is, and that also takes months to complete. We're also engaged with Rett syndrome clinicians as well as the patient community to really help us craft the right clinical trial design for our first, clinical study for NGN-401. So, as I'm sure everybody here can appreciate, if you're not thoughtful about the way you design the study, you might miss some benefits. And so we want to be really thoughtful about the way, who gets enrolled and how it's designed in order to accelerate the development of the drug as quickly as possible. And then we're also, very actively engaged with regulators, including both the US FDA as well as outside of the US. And we need to gain their feedback, and we are gaining their feedback on all the different regulatory requirements, not only because it's gene therapy, but because Stuart told you we've got this novel kind of thermostat built in, and no one's ever seen that before. So we need to make sure that we're completing all of the requirements to satisfy regulatory needs as well. So you can see it's like there's just this a lot of multiple parallel work streams that are flowing together and a large amount of moving pieces. And, all of this needs to happen before we can submit a formal application to regulators to actually gain their approval to advance into a clinical trial. So to be transparent in terms of the milestones, because it's like there's all these pieces moving all at the same time, you're not really going to hear much from us in terms of sort of a next step. We'll obviously, at additional scientific congresses, continue to, elaborate on some of the data that we put out. But in general, really the next big milestone that I think you guys will be very excited about is when we're ready to announce, when we've gotten, kind of this approval to start a clinical trial, we will make that announcement, and, you guys will hear about it right away. In terms of timing of when that is, it's just, we don't have, we're not able to provide a, robust estimate at this time. And we just want to be careful and responsible and manage expectations in the community. And the last thing I do want to say, because sort of that'll be the next question, is once we do, assuming we do, and once we do gain the necessary regulatory approvals, it's pretty common that there's additional requests and to kind of finalize and change some of the way the clinical trial is set up. That's just pretty standard, in the way that regulatory bodies kind of react. So they're like, okay, you're approved, but we want you to make these like 25 changes. And so we just want to set expectations that once we have that approval, then, there's this additional work, sort of the follow through. Then all of the clinical trial sites have their own process of getting an ethical review of the clinical trial before it begins. So that can take anywhere from 6 to 12 months, just from the regulatory approval itself to the time that you're dosing your first patient. So just to give everybody a sense of timelines. I hope that was clear.
Yeah, no, and your last point is important, because I think, there is this impression maybe that, okay, you've got FDA approval, the first patient gets dosed the next day. And that's not typically how it works.
It's not only not typical, but it's actually not, it's just not possible. And we're not there yet. I think with other types of modalities like small molecules, you can move a little bit more quickly, and it can be on the order of like 1 to 3 months. But in gene therapy, given all the complexities, because this is still a technology that is very, very novel, even just from a manufacturing perspective, it just, there's a lot of I's and I's that need to be dotted, T's that need to be crossed, and just the approvals through the clinical trial sites, because it's so new, a lot of these institutions may have very limited experience, or they just may be very cautious. And so it just takes a lot more time. And
we should be careful, right? I mean, with gene therapy, once you inject it into a person, you can't get it out. So we should be careful. And I think that should give our families peace of mind, actually, that there are a lot of eyes on it, and that there are a lot of I's that need to be dotted and T's that need to be crossed. So, we've said many times today already that the gene therapy in general is still a very new field. And these programs, these gene replacement programs, are getting a lot of scrutiny from the FDA. But despite the tough regulatory environment, your company has received FDA clearance to begin a trial for Batten's disease, CLN5, which is a type of Batten's disease. And that's a big accomplishment. So, congratulations for that. Can you share with us kind of your feelings on the current regulatory environment? Why is it so challenging? And what do you think that might mean for Rett syndrome and your program?
Yeah, I mean, I think despite some of the setbacks, which I'm going to talk to you about, because I want to make sure that everybody here has a good grounding, I have to say that everyone, all stakeholders are very excited about gene therapy, whether it's companies or families, regulatory bodies. I think everyone really is eager to get medications to patients as soon as possible. And that is palpable for us when we engage with regulators. So I think as long as you have very strong data and you've done, a lot of work, there is very strong enthusiasm. But, as we talked about, it's novel, with novelty comes surprises. And a few years ago, even lingering into today, there's a huge focus on manufacturing. If you think about it, it's just, you might not sort of realize this, but like we're actually using cells to manufacture a virus that contains this very carefully designed, MECP2 gene that sits inside this, FedEx truck, so to speak. The process of making that all happen and making all of the viral particles contain exactly what you designed, it's just not perfect. And as a result of that, you really need to know, what did what's in there? How did that compare to what was evaluated in animals, in your toxicology studies, et cetera? And so, you really needed to perform a lot of experiments in a very rigorous way. So I would say gene therapy manufacturing, you don't hear about it as much, but it's still an important piece that the FDA and others are very, very focused on. Beyond the manufacturing piece, there have been a series of adverse safety events linked to the administration of some investigational gene therapies. So, for example, the FDA, in an effort, as you pointed out, to really ensure human safety, we've just seen them be much more aggressive at pausing clinical trials at the first hint of a safety signal. Even if it's something like an abnormal liver enzyme, it's like, well, wait a minute, hold on. And they just sort of put the brakes on it. They don't want any more patients to be exposed until there's like additional mitigation, plans in place, monitoring, or whatever the specific issue is. And so, that just sometimes it's a pretty quick fix of like, okay, it'll take maybe a couple of months. In other cases, it can really put a program on significant hold. So I think, as Stuart mentioned, with all the different, all the different products in development, like there's sort of bound to be, all these diseases are different. So we're sort of bumping our toes and, hitting our heads a little bit as a field as we bump into these issues in different indications. So, in terms of Neurogene, I guess what I want everyone here to understand is that, and this may be related to the fact that I did this for a living for 13 years, or my job was to analyze, industry trends and companies in a very deep way. Like, we watch other gene therapy companies like so closely. We are really trying to learn as much as we can so that we're not, making the same, I don't know if I would call it a mistake, but just sort of, so we're getting over whatever obstacle that, other companies. So we really proactively build measures, as much as we can based on all the data that we know to mitigate safety risks. As we talked about, the obvious concern for Rett syndrome is really, kind of proving that our product isn't going to be harmful because of the gene itself. So in terms of us, we're very focused on providing a robust safety package. I think the efficacy data that was presented at ASGCT is very compelling, but it's really making sure that there's a very robust safety package underlying that to really support the overall program with regulatory agencies.
Yep, makes a lot of sense. So just to switch gears a little bit, so RSRT is spearheading a number of resources, things like the Rett Syndrome Global Registry, the Natural History Study, a digital natural history study, an effort to identify biomarkers that Stuart's involved with, objective assessments of symptoms. And can you tell us a little bit, Rachel, about why these efforts are important to all companies, including Neurogene, and how they might affect your current program and potentially future ones?
Yeah, I mean, I can't underscore enough how critical natural history data are to not just advancing, but really accelerating a program from a mouse to a human and then eventually approval. Whether it's the FDA or outside of the US, these regulatory agencies always want to know, how can you be sure that your treatment is improving disease? So, many of these early studies don't have what we call a, placebo control. And that's a whole nother, ball of wax. But as a result of that, how do you know that just, the child wouldn't have, kind of gained that skill? And so if you have a very firm grasp on the natural history, you're able to kind of say to the FDA or other regulators, see, look, like this never happens in Rett syndrome, and we're seeing it here. This is cause for real excitement. And the sooner you can point out what looks like a treatment effect, the better. So it's a tool to educate the regulatory agencies, because you all know what's going on in the disease, but how do you translate that into a body of data that you can then have a very logical, rational argument, but just sort of presentation to the regulatory bodies. I think, so maybe that that's sort of the big piece there.
Thank you. So Stuart, we know that scientific discovery is often very incremental, right, and iterative. And breakthroughs are typically the result of many years of research. I mean, your ASGCT, I think, presentation is a good example of that, right? It's like such a strong data set, but that was years and years of work, right? So, and now we're on the cusp of clinical trials for genetic-based treatments for Rett and other disorders. How do you see the landscape for genetic-based treatments for Rett progressing, and what do you hope that Neurogene's role will be in the progress?
Yeah, so I think there's obviously been an explosion in genetic therapy approaches, and the Rett Syndrome Research Trust have been amazing and sort of bringing in lots of attempts to target the genetic underlying cause of Rett syndrome and target that from multiple genetic therapy approaches. I guess from a Neurogene perspective, we sort of welcome the fact that there will be multiple approaches, and I'm sure the broader community supports this as well. And I think having multiple treatment options is important because Rett syndrome is a complex disease and it's quite individualized as well. So some individuals have more seizures and others have more motor difficulties, and it's very varied, and no two patients are identical, and therefore multiple options to address the specific needs of individual patients is something that's really important. But besides that, the sort of broader concept is that the more shots on goal you have, the more likely it is that one or more will succeed, and you will have genuine, credible treatment options that are available for people with Rett syndrome. So overall, we welcome the fact that Rett Syndrome Research Trust are sort of really pushing targeting the genetic therapy approach from all directions. It's really
great.
welcome.
We agree. All right. I think we're going to move to some Q&A now. These are questions that have been submitted before the webinar. So the one question that's on everybody's mind is, and we've already touched on it during the webinar, but I think it's important, and so we received it from lots of different people, so it's probably worth repeating. So can you summarize what phase the research is in, and what is the timing for a clinical trial?
Yeah, so just to reiterate, we are in the preclinical stage, which means that we're in animals. We're not ready for humans yet. There's, as I sort of went to lengths to describe, but I won't rehash everything. Stuart shared some of those animal results at the big genetics meeting called ASGCT in May. And you can find, sort of just a summary of those results in our press release if you go to our website at neurogene.com. In terms of the timing, it really is too early to make a statement. We are still conducting additional animal studies and obviously need to complete all of that, including regulatory interactions before starting a clinical trial. So we are working as quickly as we can, but I do want to make sure that people don't think there's clinical trial announcement tomorrow or anything like that.
Yep. The other question we received a lot from families from all over the world. We got questions from Africa, South America, Taiwan, and it has to do with, is there an opportunity for research in those regions? Will there be a clinical trial in my country? Can you address that?
Yeah, and we very much appreciate that there's interest from countries around the world, and certainly if it was my daughter, I would go to the most remote place, in order to get my child enrolled in a clinical trial if I thought it could help her. At this point, we do not yet know where the clinical trial sites may be. We're certainly exploring clinical trial sites in the US as well as outside of the US, but no decisions have been made at this time.
Okay. Stuart, we got a lot of questions about the exact technology, and you've already described it on our call today, on our webinar. But maybe you could summarize once again quickly why you're using full-length human gene and how the exact technology works.
Basically, Neurogene is sort of committed to sort of innovating beyond the sort of first generation gene therapy approach, which is, taking a gene, putting it in a virus and just delivering that in a sort of uncontrolled sense. And the reason for this is that, as many of you will know, a key concern with gene therapy for Rett syndrome specifically is that the effective or the efficacious dose that's likely to be therapeutic often sort of overlaps with a dose that is producing too much MECP2 and can drive adverse effects or toxicity. So what you're really trying to do is sort of thread the needle in that you're delivering enough protein to cells that lack functional MECP2 to have a therapeutic effect, but without sort of driving the system too far and generating too much expression in cells that express a healthy copy of the gene. So the technology is basically a circuit or a thermostat. It's a self-contained thermostat that tries to control that level to stop it going too high. So it's almost an exact analogy of a thermostat. If you set the temperature to 72 and it goes above because you're delivering too much, then it will just turn it back down again and moderate. In terms of the fact that we're delivering the full MECP2 gene, as I mentioned before, MECP2 is a highly conserved gene. So if you look at so in primates, for instance, it's almost completely identical. So out of the all the amino acids that make up the protein, there's a single very conservative change. And as you go back in the evolutionary tree, it's highly conserved MECP2, which suggests that even making slight changes can be deleterious. So we just want to put back in the nervous system the version of the protein that's normally there doing its function. So that's really the what we're trying to achieve here.
Great. Rachel, families of boys with Rett syndrome, they want to know if Neurogene plans to include them in studies.
Yeah, at this point, I do want to be transparent that we are planning to include females only in our first human clinical trial, but certainly, future expansion into different types of patients, will be dependent on the findings in our first trial. So I think at this point in time, the answer is no, but that doesn't preclude, future clinical trials.
Okay. And will prior participation in any other gene therapy trials preclude participation in a Neurogene trial?
Yeah, so
we've been getting.
most comments about it, are the clinical trial design. I'm sort of saying I don't want to make any comments, but this one I felt it's really important to address this. The answer is absolutely 100% no. You cannot, kind of hop from one gene therapy trial into another, and the reason is that just as an industry, we haven't yet figured out how to redose in gene therapy. So unfortunately, there's just not an established safe way to, receive one gene therapy and then get another one afterwards. This is absolutely an active area of research. So I don't think this will be a true statement forever. But certainly over the foreseeable future, I think it's going to take several years to find that solution.
Yeah, but so it'll be figured out, and potentially people can receive multiple doses, but not in the setting of a clinical trial, right? Because you wouldn't know the improvements. Were they from the first trial you were in, or are they from the second trial? So usually you want to be right, have patients that are, what's the term they use, naive to genetic-based treatments.
Treatment naive. Yeah, I think it's going to take some time. I think if I had to map this out, I would say, if anything, you could say, well, let's just say maybe the gene therapy, effects are waning over time. If you found that, then maybe the company who was sponsoring that particular gene therapy would have done enough work to sort of support a redosing, when that gets figured out, and then you could be eligible to kind of get retreated with the same. When you start talking about mixing and matching, then you're layering in an extra layer of confounding is what you're talking about. So yes, that's right.
That's a way in the head kind of thing. So
very much in the future.
First step first, we need to get the first trial going.
That's right.
That's kind of my philosophy, in general. With these are big problems. It's like, tackle one big problem at a time.
That's right. Let's get it.
We also received a lot of questions, Rachel, about age ranges for clinical trials. Anything you can share about that?
I would love to share with you our thinking, but to be completely honest with you, it's like until the regulators put their stamp of approval, it doesn't matter, what we think. So we don't have a definitive answer on the specifics of who's going to be eligible. So we're not going to be able to comment at this time.
Yeah. I think these questions we've really kind of addressed already. We've addressed the timing of a clinical trial, when the potential FDA approval may take place, we don't know yet. Probability of success. That's hard to know, but would you like to take a stab at that?
Yeah, when I saw the question, so I very much appreciate it, again, because my past analyst days, probability of success is something that it's language that I appreciate and respect. But I think, the question really means, different things to different people, because how are you defining success when you're talking about a treatment? Gene therapy is obviously still young as a treatment modality, and there's no approved treatment. So for us, what I can say is we are bringing the best science of the day forward. And, we're actually even creating it, right? You heard it's coming from Stuart's lab. And so we are going to provide as much evidence as possible to really demonstrate to experts, regulators, the community, all of the data in animals. But at the end of the day, and we're super excited, like, let me be clear, like, I couldn't be happier with, where we are. And so we hope that this treatment, NGN-401, really has a meaningful impact on the disease course. But the reality is, until we actually get some, have some data in actual people, it's really not possible at this stage to determine probability of success. And so I just want to, again, sort of express my enthusiasm, but temper that with, we don't know what we don't know at this point.
Fair enough. Stuart, we got some questions about the exact technology, and is it relevant for any kind of MECP2 mutation? So that's the first part of the question. The second part is, can you comment again on ICV versus intravenous route of administration?
Sure. So I guess the first question relates to mutations, and so I think, as many of you know, there are lots of different mutations in MECP2 that lead to Rett syndrome, and it's quite complicated. So there is some correlation between the type of mutation and the severity of Rett syndrome, but it's not a perfect correlation. There's a lot of variation, and it's also important to keep in mind that in girls with Rett syndrome, that half of the cells actually express normal levels of functional MECP2, and this is why the exact technology is sort of constraining or preventing this sort of high overexpression. But fundamentally, anyone with a Rett syndrome diagnosis will have a fairly substantial reduction in the activity or function of MECP2, and therefore should benefit from a restoration approach like this. So our vision is that NGN-401 could be used regardless of the MECP2 mutation, but ultimately it will not be for us to decide that, because a key is that the regulators will have a view on this, and so that's really dependent on what the regulatory bodies view as acceptable in the first trials.
And the ICB, I think you've already answered that. I mean, you've got data to show that you get best biodistribution.
That's correct. Yeah, I mean, with systemic delivery of the virus, there's very little gets into the brain. So this is why we're having to use this less familiar, what's called a route of administration, but it's really to maximize the likely delivery of the therapeutic to where it needs to go.
Great. So we're coming up to the hour mark, and I want to thank you so much for spending time with us today. You've got the undivided attention here of the people that are on the call, which are mostly parents of children with Rett, and then people that will watch the recording. What would you like to say to them? Parents, grandparents, anybody who loves a child with Rett syndrome. Any words to them specifically, Rachel?
All right. Well, with that intro, I'm gonna try not to cry. I guess what I want to say is we work with a sense of urgency every day. We know that you've been waiting, some of you, for a very, very long time for a treatment with potential to improve, the lives of your children and your families. So that's sort of the most important thing that I could possibly say. The concept behind gene therapy really sounds very, very simple, but unfortunately, simple does not equal easy. And I've tried to give you a flavor of the various complexities. So we do ask for your patience as we continue to very rigorously work through these, various work streams to pull everything together to put NGN-401, into patients. I know I'm not going to go through all the details, the foundation can provide, but we do have on our website, access to our patient advocacy team. And, there's information there. We have a letter to the Rett, syndrome community. And certainly, you can follow us on, our Facebook page and, check in with our websites to look for additional information.
Thank you, Rachel. And my colleague Emily is putting into the chat the various links to the items that Rachel just mentioned. Stuart, you've met a lot of individuals with Rett over the years, including my daughter, Chelsea. Anything you'd like to say to the families?
Yeah, I mean, Monica, I'd really just like to sort of mirror what Rachel has said, and thank the community for their patience. Rachel sort of emphasized how urgency is really central to our sort of mission, but we appreciate that the science can be frustratingly slow. It's much slower than everyone would like. It's just, unfortunately, that's just the way science moves quite often, and we realize this, but we appreciate that many of the families have been following progress over many years, and we thank them for everyone for their patience and also for their support. Yeah. Great.
Thank you both, and we appreciate it. I think I speak on behalf of every family to say that we wish you Godspeed, and we're here for you. The community is here to help. I know that you'll be needing more information about symptoms and prioritizing symptoms and what families think, we're here to help. RSRT is here to help as well. And have a great summer. And I'm going to take you up, Rachel, on that webinar on manufacturing.
I know, when it came out of my mouth,
I wrote it down.
Like, oh, no. I went off script. I'm not supposed to do that.
Well, we'll see. But, you know, there's other interesting topics, right, to discuss with gene therapy. So we'll try to keep the community, you know, updated as much as we can, as much as you're able to do. All right. Thank you, everybody. Happy 4th of July to our families in the US. Thanks for joining us. Bye-bye.
Bye.